Hi, I'm Emilio Penny, and I'm going to be stating some interesting facts about this molecule called Juglone. Its chemical formula is C10H6O3, and it belongs to a class of organic compounds known as naphthoquinones, which consist of a benzene ring linearly fused to a benzene 1,4-dione, also known as a quinone. It can be found in many different foods, but it is mainly found in black walnut trees and its roots. It's an allelopathic compound, which means that the tree secretes this compound into the soil, so it acts as a natural herbicide and protector of walnut trees. It is eventually leaked out in the soil and also in the new seeds to deter insects from eating them. Some of its physical characteristics: it weighs 174.15 grams per mole. Its melting point is 155 degrees Celsius, while its boiling point is way above that at 385.80 degrees Celsius. Its water solubility is 5,121 milligrams per liter at 25 degrees Celsius. It can be classified as an aromatic homopolycyclic compound. And although no enantiomer has been found, there are many different juglone derivatives depending on the position of their substituents. Inside the tree, it is in a form called pre-juglone, and it is safe. But once pre-juglone touches air, it becomes oxidized and turns into juglone, which we know as a sap-like toxic substance. The fruit, leaves, and roots of black walnut trees all contain juglone, and they can have a devastating impact on the roots of neighboring plants. In humans, ingesting even a small amount of pure juglone can cause a serious poisoning effect. Although it's not established to be flammable, it is toxic to inhale, touch, and ingest, and it is very toxic to aquatic life. Because of the antioxidant capacities of phenolithic compounds, juglone may serve to combat oxidative stress, thereby protecting against the development of various diseases and aging processes. Most notable is its juglone's inhibition of PIN1 that could regulate the phosphorylation of TAU, implicating potential effects of juglone in helping Alzheimer's disease research. Juglone also activates mitogen-activated protein kinases that could promote cell survival, thereby protecting against conditions such as cardiac injury. As stated before, it is very toxic to humans if ingested, touched, or inhaled. Normal fibroblasts were found to be especially sensitive to juglone and lost all viability primarily through rapid apoptic and necrotic responses. This may have been triggered by DNA damage since juglone induced a rapid and strong phosphorylation of H2AX in all phases of the cell cycle. Juglone also inhibits the mRNA synthesis in human fibroblasts in a dose-dependent manner. All in all, juglone can be stated that it induces apoptosis of most human cells. The harmful effects of walnut populations have been observed since before 1850, and since then have been used to fish efficiently well. In 1850, two men, A. Vogel Jr. and C. Reichhauser, were able to isolate the compound from a walnut tree in 1851. The compound was known as mucin at the time. Then juglone was synthesized and characterized for the first time in 1887 by A. Bernstein and A. Semper. Later, in the 1900s, it was confirmed to be toxic to plant life as well. It was mainly found in walnut trees, their roots, leaves, and fruit as well. But it is not rare. But it can also not be classified as extremely abundant. Juglone, even though it's a toxin that's not very renowned, nevertheless seems to be a rising star in recent chemical studies. This allelopathic chemical has been used for many centuries now in a variety of ways and treatments. The natural herbicide and pesticide was noticed in the 1800s in walnut trees and their surrounding areas. This now can lead to a new natural, less harmful herbicide. Juglone's toxicity has also been noticed in earlier centuries as an effective aquatic toxin to fish with. Recently, its toxins have been narrowed down to cause cell apoptosis and can now be used to help cancer and Alzheimer's research. Although its history is not the most humane or relevant, Juglone nevertheless presents us with a lot of future potential, and it can help us discover better, more natural, and less expensive medicine. Thank you for your time.